Hey, welcome. You ever stop and wonder how two things can seem identical on paper, but be totally different in reality? Well, that's a huge puzzle in chemistry, and that's exactly what we're diving into today. We're going to look at how molecules with the exact same chemical formula could be completely different substances. It's wild. So here's how we're going to break this down. First, we'll tackle that central mystery. Same formula, different molecule. Then we'll get into the three-Lee twist that honestly explains everything. We'll even pinpoint the exact atom that makes it all possible, meet the different members of this molecular family, and finally, and this is the really cool part, we'll see why a tiny change in shape has huge consequences for our own bodies. All right, let's jump right in. We're starting with a real head-scratcher that's at the heart of this whole thing. So look at this. You've got a chemical formula, C6H12O6. Think of it like a recipe, right? 6 carbons, 12 hydrogens, 6 oxygens. You'd think if you put those ingredients together, you'd get the same cake every single time. But what if you don't? How is that even possible? Okay, check this out. On the left, we have D-glucose, our body's favorite fuel. And on the right, D-galactose, which you get from dairy. They both have that same C6H12O6 formula. I mean, look at them. They're almost identical. But here's the thing. Our bodies treat them completely differently. So what's the catch? What's the hidden difference? Well, the answer isn't on a flat piece of paper. It's in the real three-dimensional world. And this is where things get really interesting. See, it's not about what they're made of. We already know the ingredients are the same. It's all about how they're built in 3D space. The assembly is everything. And this whole idea has a name. It's called stereoisomerism. Basically, it's when you have molecules with the same formula, the same atom-to-atom connections, but they have a totally different 3D arrangement. The best way to think about it is with your hands. Go ahead, look at them. They're mirror images of each other, right? But they are not identical. You can't perfectly stack your left hand on top of your right, and you definitely can't put a left-handed glove on your right hand. That's what we mean. They're related, but they're distinct. Yeah, and this quote from our source material just nails it. The difference lies entirely in how those groups are positioned in three-dimensional space around something called the chiral center. So, okay, what's a chiral center? So what is this chiral center the quote just mentioned? Well, it's the linchpin for this entire concept. It all comes down to a very, very special kind of carbon atom. And here it is. A chiral carbon atom is simply a carbon atom that's bonded to 4 different groups or atoms. That's the key. 4 different things attached to it. This is what creates the opportunity for different 3D arrangements. It's the pivot point for all this variety. This is a perfect example. Look at this molecule, glyceraldehyde. See that carbon with the little asterisk? That's our chiral carbon. Now, don't worry about memorizing the whole structure. Just notice what's attached to it. You've got an H, an OH, a CHO, and a CH2OH. 4 completely different groups. And that's what creates the center of asymmetry. Okay, so now that we know the how and the where it happens in 3D space around a chiral carbon, let's get to the what. It's time to meet the different members of the isomer family and see how they relate to each other. We're going to apply this right back to our original mystery with glucose. For today, we're really going to focus on 2 key relationships here. First up, you have enantiomers. These are the perfect non-superimposable mirror images we talked about, just like your hands. Every single chiral center is a mirror reflection of the other. But then you have epimers, and these are way more subtle. They are almost identical twins, but they differ in the 3D arrangement around only one single chiral carbon. So one is a total mirror image, and the other is a twin with just one tiny little difference. All right, let's go back to our friend D-glucose. To really get what an epimer is, I want you to look closely at the 4th carbon atom from the top. See where that OH group is? Just lock that picture in your mind. Okay, now here's D-galactose. Take a look. Everything else is exactly the same as glucose, except for carbon number 4. See how the OH group has flipped to the other side? That one single change is what makes it an epimer of glucose. And we're not done yet. Here's D-mannose. Now, if you compare this one to glucose, carbon 4 is back in the same position, but look at carbon number 2. That's where the flip happened this time. So, mannose is another epimer of glucose. Isn't that wild? Just one little flip and you've got a whole new molecule. So, this naturally leads to a really fascinating question. We've just seen that flipping one chiral carbon creates a whole new molecule. Well, glucose doesn't just have one, it has 4 of these chiral carbons. So just how many different versions of this molecule are even possible?
Well, luckily, there's a really simple formula for this. You just take 2 to the power of n, where n is the number of chiral carbons in the molecule.
So for glucose, with its 4 chiral carbons, that means there are 16 possible stereoisomers. That is 16 totally different molecules, all with the exact same C6H12O6 formula. Wow. Let's just quickly break that down so it sticks. We take 2 to the power of 4, because glucose has 4 chiral carbons. That gives us 16 total possibilities. And since enantiomers, those mirror images, always come in pairs, that means we have 8 pairs of these molecular twins.
16 different versions from one single formula. That's kind of mind-boggling, right? But you might be wondering, does this actually matter in the real world? And the answer is a huge yes. In fact, it matters inside every single one of us, every single day.
And this is really the key takeaway from all of this. Our bodies are incredibly picky. As this quote points out, we almost exclusively use what's called the D configuration for sugars. Our cells can actually tell the difference between D-glucose and its perfect mirror image L-glucose. So why? It's because our enzymes, the little machines in our cells that process these sugars, are also 3D. They have these perfectly shaped pockets, and only the D form can fit into them. It's like a key fitting into a lock. The L form, it just won't fit. So we can't use it for energy. The 3D shape is literally everything.
And that really leaves us with a final thought to chew on. If flipping a single atom in 3D space can completely change a molecule's role in our own bodies, what other invisible differences are out there shaping our world? It's just a powerful reminder that sometimes the smallest, most hidden details can make all of the difference.